Thank you for joining us for today's seminar series as part of the Coulter Foundation Biomedical Engineering Seminar Series. So let's get started. It is my distinct pleasure to introduce Dr. Adrian Chester, who joins us from the Imperial College in the London, UK. He will be providing our seminar today, as well as he has a schedule with several of our faculty and students later today. So Dr. Chester, he essentially completed his PhD and with under the supervision of the eminent surgeon, Sir Magdi Yaqub. And he has since established a laboratory at Harefield dedicated to looking at vascular mechanisms. And his more recent work and research investigates valvular cells in heart valve function, and specifically looking at aortic valve calcification. And he also does quite a bit of research looking at tissue engineering of valves, which is actually the topic of his seminar talk today. Of course, tissue engineered heart valves in particular hits home for me because that's also a active research area in my laboratory. And so I'm really eager to, hear him talk about this. And of course, in a way, it's nice. I was talking to Dr. Chester, and it seems like these type of Zoom seminars are advantageous in that, otherwise under normal circumstances, we may not have had an opportunity to hear each other, across the pond like this. But this is great. And again, it's our pleasure to have Dr. Chester here with us. Without further ado, let us welcome Dr. Chester, and his topic of his talk is biological complexity of the aortic valve, what is relevant for tissue engineered heart valves. Dr. Chester, it's all yours.
Thank you. Sharon, thank you for the kind introduction. Okay, so this morning for you, the afternoon for me, I'm going to talk about some of our work on the biology of heart valves and the complexity and the role of cells, and then reflect on, out of what we know for heart valve biology, what's relevant for tissue engineering heart valves, and share with you some of our experiences in the development of our tissue engineered valve. So, of course, this month in the USA and in the UK is Heart Month. So I've donned a red shirt, the only one I've got, in honor of this. And I believe you have some other heart-centered talks during this month. And I looked at the program you have lined up, and you have some excellent speakers on heart valve tissue engineering and heart valve biomechanics. And in some respects, it then fits that my talk is a little bit of a warm-up for some of the talks to follow, giving you an insight into the mechanisms of what the cells do in heart valves, and again, their relevance to tissue engineering. So here is the heart, and for just anyone who might not be familiar with the anatomy of the heart, the four valves within the heart are slightly different in nature. You have the two AV valves, which have a sort of a parachute type mechanism for closure, and the semilunar valve seen here, the pulmonary valve easily seen in this diagram, which is in the anatomically similar to the aortic valve, which is just shown behind this in this picture. And these two valves particularly, while similar in structure, the aortic valve is the valve that becomes affected most by disease or congenital abdominal malformations. It is therefore the subject of surgical replacement, and therefore the focus of many tissue engineering projects. So we're going to focus on how this valve works in terms of the complexity of its structure-function relationship, and how that may be relevant to tissue engineering. So a few things to consider in relevance to the challenge in tissue engineering a heart valve, and you're trying to recapitulate something that is a highly dynamic and continually moving structure that's in perhaps one of the harshest mechanical environments within the body. It's exposed to diastolic pressure on a cyclical basis, and it's been calculated, I quote this number quite a lot, that in an average lifetime, which is nowadays measured at 75 years, but it's probably more, that your valve will open and close 3 billion times. And in most of us, it will do so without malfunction at all. But you have to remember this number is calculated on a heart rate of about 75 beats per minute and doesn't assume that your heart rate may vary over that time. And of course, your valve manages to function under very deeply differing hemodynamic conditions that might occur during exercise, for example. So therefore, exploring what I would term the structure, biology, function relationship is what is important in understanding what needs to be regained or remade in a tissue engineered tissue in heart valve. So just for anyone who may not have seen, this is what a human heart valve looks like. And we can see the 3 cusps here, which are very thin, almost translucent pieces of tissue. And they're attached to what forms a crown-shaped annulus. So I've gone forward here. And then there's a ridge, which is even hard to see by eye, that are called the sinotubular junction, which marks the boundary between the aortic valve and the ascending aorta. And in this picture, you can see the different structure of the anterior leaflet of the mitral valve. So what happens to valves in old age in a certain subset of people is they form calcified lesions on them, and they become thickened and therefore immobile and are in need of surgical replacement. And in the diagram to the right, you can see what's termed a bicuspid valve. So this only has 2 cusps. And depending on the configuration of these 2 cusps, these patients develop exactly the same sort of disease, but at a much younger age. And these people are the candidates for a tissue engineered valve, since as we will hear shortly, that the current heart valve substitutes have a number of shortcomings that mean that if you're of a younger age, and I'm talking anything from early 20s to 40s, the normal valves present a number of problems for you if you're going to have your valve replaced. So the current heart valve substitutes that are in use and on the market fall into sort of 3 main categories. The mechanical metal valves, which would last you a lifetime, no doubt about that, except you then have to take blood thinners for the rest of your life. And that then comes with a certain amount of inconvenience, but also a certain amount of risk from thromboembolic events and risks of bleeding. And then you have tissue valves, which are normally glutaraldehyde fixed porcine or bovine tissue sewed into stented rings. And these are typically given to older patients, and their life or longevity is something between 10 to 15 years before they undergo like a degenerative disease. And then to the right-hand side, you see more anatomically intact tissues. And then to this, you could add homografts, which are human valves that have either been cryopreserved or fixed, and then try to recapitulate some of the anatomy of the heart. But the one thing all these 3 types of heart valve have in common is none of them have living cells. And the only valve substitute that has a living cell is the so-called Ross operation, or the pulmonary autograft. And just to briefly explain this, that this is quite a rare operation and is only done by, if you like, surgeons who have an expertise in actually performing this type of surgery. And what you do is you remove the pulmonary valve from the patient, which is normal and undiseased, and you put it into the aortic position and replace the pulmonary valve with an autograft, like we just seen. So the autograft in the pulmonary position will last because this is a low pressure situation and is without any great wear and tear. And the pulmonary valve that belongs to that person sits here, adapts to its new situation, and like any other valve substitute, it contains living cells. And in Professor Yakub's series that he published relatively recently in the Lancet, he showed that the autograft shown in green was superior to other human valves implanted, so-called homografts, which don't contain living cells. But perhaps the most impressive thing about this data is that the dotted line underneath the green line is the incidence of death in an age-matched population, showing that the pulmonary autograft is like having a normal valve. So this is a proof of principle that if you can give somebody a valve with living cells, and those cells can recapitulate the function of heart valve cells, then your valve should last you the rest of your life. So as far as heart valve tissue engineering goes, this can be divided broadly into 3 different strategies. First strategy, and probably the one that most people started out investigating, certainly true for our experience, was so-called in vitro tissue engineering, where following the normal paradigm of tissue engineering, you're taking scaffolds, mixing these with a cell source of your choice, making a cellularized scaffold or valve, bioconditioning this and some sort of bioreactor prior to implantation. Other strategies, certainly this one's not as popular or as well pursued, would involve implanting your scaffold into an animal, letting it become cellularized, and then implanting it. And the more popular and currently much of the focus of our work and the work of other groups is so-called in situ tissue engineering, which can either be a scaffold which is cellularized, then decellularized, so you get the cells to lay down extracellular matrix onto your scaffold, which is then implanted, or you take a scaffold which either can or may not, is or isn't decorated with biomolecules to make cells recognize certain integrin-like molecules or peptides that will confer cellularization by desired cell phenotypes. And this then biofunctionalized scaffold is then implanted. So these are the 3 main strategies for tissue engineering, and we'll reflect back on these towards the end of the talk to see how what we've learned and know about tissue engineering or valve biology and how it can be applied to these strategies. So in terms of the heart valve, a living heart valve, we'll cover 5 main areas: the complexity of the structure-function relationship and what it means to heart valve function, the importance of valve cells with particular reference to the role of the endothelial cells, and interactions between cells and the extracellular matrix. And lastly, that leads into mechanisms of mechanotransduction. So in terms of the complexity of a heart valve function, I've got 2 examples of this for you. So if you focus here in the first circle, these 2 parallel lines just mark 2 points in time. And this is the change in aortic pressure. And the pressure goes up after the second line. And if you look at the bottom line, this is aortic flow. And then that comes slightly after, or as with the change in pressure. But if you look in between, you have aortic valve orifice area, and this is the valve opening. And in the time duration, which is here, is probably as milliseconds, is the valve actually begins to open before there's any change in aortic pressure and before there's any forward flow of blood from the vent through the valve. So in reality, the valve is anticipating ejection of the left ventricle and beginning to open, ready for the forward flow of blood. So i.e., it is not just a passive conduit that responds entirely to the motion of the blood from the ventricle. And it achieves some of these effects by the fact that it's a highly dynamic structure, even at the base of the annulus or at the level of the sinotubular junction. There are specific movements that occur throughout the isovolumic contraction and the ejection of the blood, whereby the levels of the annulus either move apart or together, and the level at the sinotubular junction also move. There is also some rotational changes, and this dynamic function of the aortic root, which refers to the whole annulus up to the sinotubular junction, this dynamic function led to Professor Jakob's paper where he termed the phrase dynamism and crosstalk, i.e. there's crosstalk between different structures of the aortic root, and dynamic function of it means that it optimizes its opening and closing throughout the cardiac cycle. So if we move on and look at some of the cells that can be found in heart valves and understand a little bit about what their function may be and why they're important to the structure and to the durability and longevity of the valve cusp. So a valve cusp has a very characteristic tri-layered function where you have a ventricularis, which has mainly elastin in it, spongiosa with the glycosaminoglycans, and collagen-rich fibrosa. And both sides are covered, as is the whole of the vascular system, with a continuous layer of endothelial cells. So here's some immunohistochemistry of valve cusps, showing firstly the presence of the endothelial cells on both sides of a valve, and markers of fibroblast-like cells with respect to vimentin and fibroblast surface antigen, and evidence of expression of the enzyme that's responsible for collagen synthesis, proline 4-hydroxylase. Interestingly, some of the cells exhibit contractile proteins that may be present in myofibroblast cells or even perhaps smooth muscle cells. And we'll explore a little bit later on exactly what the function of those cells is. So returns to valve interstitial cells. What are their principal relevant components in terms of function of the valve and what is relevant to tissue engineering? Is their ability in response to stretch to synthesize and release collagen. So in the bottom panel here, we see the stretch dependent, levels of stretch dependent increase in collagen synthesis. And this collagen is incorporated into the cells, whereas the top panel show collagen released into the media. So the valve interstitial cells are responsible for collagen synthesis, which helps with the longevity and the homeostasis of the valve. This is confirmed in the study by the Balacharan investigators, which showed similar effects for collagen and the fact that stretch also regulates GAGs, but with no effect on elastin. Stretch can also upregulate levels of smooth muscle alpha-actin, which is indicative again of presence of myofibroblasts. So there are some myofibroblasts in healthy cells. These tend to reduce immediately after birth, but then may increase as markers of disease later in life, but they also play a role in healthy valves. And in fact, this was the first data that got me personally involved with heart valves. As Sharon alluded to in his introduction, my initial work focused on vascular mechanisms, and I did a lot of work in isolated tissue baths. And one of the researchers who was looking at heart valve cells and characterizing them came to me and he said that he had seen that he could get a calcium signal from cells when he added things like endothelin and 5-HT. And he knew from his own experience in working with blood vessels that both these agents could produce contraction of vascular ring segments. So we embarked on a study whereby we took isolated porcine valve cusps, put them in a tissue bath, and I started then adding various contractile agents and managed to show that there was a whole repertoire of receptors present on porcine valve cusps that could induce contraction. One thing I noticed compared to ring segments, these contractions were quite small. And at the time, I had no real notion as to what the relevance of these contractions were to heart valves. They occurred over a relatively slow period of time, which meant that they were not involved in the beat-to-beat opening and closing of the valve. And at the time, I postulated, albeit to myself, that perhaps there was some sort of valve tone in existence. Just to further elucidate the complexity of the role of contraction is that valve cells also contain a neuronal supply, and this could be shown to be parasympathetic, sympathetic, or peptidergic. So I then also took valve segments in exactly the same manner and electrically stimulated them and witnessed both shown in this trace contractile responses to electrical stimulation, which is shown in the dose response curve in the lower panel. And these were inhibited by a blocker of neuronal transmission, namely TTX. You could also see, although not shown on this slide, relaxations to neuronal stimulation, which turned out and were shown to be due to nitrogen nerves releasing nitric oxide. So in order to try to pursue the concept of whether these contractile mechanisms had a relevance to valve tone, as it were, with the help of then teaming up with bioengineers, and this was for me a new excursion in scientific methods, was we took a biaxial mechanical tester, and rather than do this in the open air, we then built a tissue bath essentially around this, so we could keep the tissue alive as it was in a organ bath. But yet, instead of doing contraction experiments, we endeavored to do uniaxial testing and constructed stress strain curves in the radial and the circumferential direction, and then measured the stiffness of the tissue, or the Young's modulus. And what we were able to show, I'll talk you through this slide, is with potassium chloride, which is a non-specific chemical way of making muscle cells contract, is that we could see that there was a 15 to 20% increase in the stiffness in the circumferential and a slightly lesser increase in stiffness in the radial direction of valve tissue. Conversely, if you added sodium nitroprusside, which is an NO donor, and you would expect nitric oxide to relax the tissue, you saw the reverse, whereby sodium nitroprusside produced a decrease in stiffness. And then when you use endogenous, or you add mediators that are experienced within the circulation, namely endothelin 1, which is a peptide released by endothelial cells, this produced a larger increase in stiffness, and 5-HT, which in valve cells releases nitric oxide, this reduced the stiffness, again, by a greater percentage than the sodium nitroprusside in both the circumferential and radial directions. And therefore, we were confident that, in fact, valve tissue is under the influence of vasoconstrictor stimuli, which may be either in the blood or released by the endothelial cells in order to optimize the tension under which they operate. So addressing more specifically now the role of the valve endothelium, which I alluded to just there with the effect of 5-HT, but just to refresh anybody's minds as to the importance of the endothelium in vascular tissue, where it has been widely studied, the endothelial cells, which serve as the interface between the vessel wall and the blood, in stimulation by the flow of the blood across the surface into the endothelial cells, release nitric oxide, which diffuses to produce relaxation via a mechanism whereby it increases cyclic GMP. The endothelium and NO can also regulate platelet aggregation and the expression of adhesion molecules. So with respect to our experiments with 5-HT, both here in the radial and circumferential directions, adding increasing doses of 5-HT caused a reduction in tension. And just to show that this was a nitric oxide effect, the effect was turned from a relaxation to a contraction by a co-incubation with the nitric oxide synthase inhibitor L-name, an effect again mimicked by intentional removal of the endothelial cells, just validating that the 5-HT effect was indeed mediated by the endothelium. Now, in terms of the valve is a very interesting structure with respect to the flow over the surfaces of either side of the valve. So on the ventricular surface, you can see that you get high laminar shear flow, which is indicative of atheroprotected areas in the vasculature, whereas the aortic surfaces, when the valve closes, see a disturbed low shear pattern of flow as the blood recirculates around the sinuses. And low patterns of flow are indicative in the vasculature, or low patterns of shear stress, of being atherosensitive or atheroprone areas. And indeed, calcification of valves occurs in these areas. So we wanted to look at what protective role the endothelium may have under these conditions. And in conjunction or in collaboration with Jonathan Butcher's group, we looked at induction of calcification by osteogenic media, by counting calcified nodules in samples of valve. And when we did this experiment in the presence of L-name, the numbers of calcified nodules increased principally in the fibrosa, so on the aortic side of the valve where calcification normally found, and also in the ventricularis side, demonstrating that NO released from the endothelium on the valve has a protective effect. And then just some of our recent data, just to add to that complexity of the protective effect offered by nitric oxide. We recently looked at the expression of phosphodiesterase enzymes. So phosphodiesterases will break down the second messenger for nitric oxide, namely cyclic GMP, and limit its effect. So phosphodiesterase inhibitors promote the effect of NO. So, and we found that in calcified valves, a number of different isoforms of this enzyme were all increased in the presence of calcification compared to non-calcified valves. So this would have the effect of reducing the bioavailability of NO because you're having more breakdown of its second messenger. And we need to pursue further as to whether this increase in these enzymes, at what stage this occurs, and whether it may be responsible for the reduction in the effect of NO and the progression of the calcifying responses. We've also seen in a site-specific way that the endothelium can regulate the extracellular matrix. So in these experiments, intact valve samples were put into a cone and plate machine and exposed to aortic conditions of flow and ventricular conditions of flow. And while this is quite complex, but to summarize, essentially what you see is that the aortic and the ventricular sides of the tissue respond in slightly different ways given their different patterns of flow, and that the extracellular matrix, namely collagen synthesis particularly, and GAGs, are regulated by the endothelium in response to flow. So moving on to how the cells then interact with the extracellular matrix. And this step is important with respect to tissue engineering because you're replacing the extracellular matrix with your scaffold material. So how it ultimately interacts with the scaffold material is what you want to be able to recapitulate is how they interact with the extracellular matrix. And in fact, the extracellular matrix in some way can give instructive signals to the cells to respond in a certain way. And quite some time ago, we looked at the expression of a whole range of different integrin molecules, which are responsible for binding cells to the matrix, and show there was a whole repertoire of integrin molecules, some underexpressed, the ones that were arrows were more highly expressed in terms of the aortic valve, but also the other three valve types. And we focused some studies on the alpha-2 beta-1 integrin, since this is primarily the collagen binding integrin in heart valves. And we could see here expression of that integrin in this fluorescent picture compared to expression of smooth muscle alpha actin. And we looked at the role of this integrin in mediating contraction of collagen gels. So when cells were added to gels, they produced a reduction in gel size, seen here. And when this was their incubation occurred in the presence of blocking antibodies for alpha-2 beta-1 integrin, this level of contraction was partially reversed. So we also went on to look at the role of focal adhesion kinase, one of the signaling molecules involved in signaling induced by adhesion by integrin binding. And similar to the effect of the blocking antibody, a focal adhesion kinase inhibitor also blocked collagen gel contraction. We then looked at contraction of valve cell leaflets in response to endothelium 1, and both the blocking antibody and focal adhesion kinase inhibitor inhibited the contractile response to endothelium, illustrating nicely the role of linking cells to the matrix in order to produce a biological effect. And that leads on quite nicely to mechanotransduction, because cells, as we started off by looking at the effect of stretch and collagen synthesis, cells respond to the mechanical environment in which the valves operate. And there are a whole range of potential mechanosensors, and I've just touched on integrins and perhaps most widely studied, but other connective molecules within cells are important. There's been a lot of focus recently on cilia, particularly in mitral valves, and the glycocalyx, which is a lining of proteoglycan molecules present on the endothelium for sensing responses of flow. But we've recently looked at mechanosensitive ion channels, and I'll show you a little bit of data just about how they are expressed in different valve cell types and some of the functional roles we've been able to identify for these cells. So we looked at a whole range of potassium sensitive and calcium sensitive mechanosensitive channels, the TRP channels and the potassium channels TREC1 and KIR 6.1, and showing that you can get variable, or the expression of these channels varies according to the phenotype of the valve cell. So here we've got valve cells that are of a fibroblast phenotype, a myofibroblast phenotype, and even an osteoblast phenotype. These are driven to become osteoblasts by incubating in osteogenic media for 3 weeks. And this changes the responsiveness of the cell, or the potential responsiveness of the cells to mechanical force or sensing by these mechanosensitive ion channels. And we were able to show that an inhibitor of TRPV4 channels was responsible for collagen 1 synthesis in response to stretch. These other streptomycin is a non-specific channel inhibitor, and these significant, but there was some degree of inhibition, and the same for the TRPC6/3 inhibitor. But in terms of Vic migration, we also looked at this by the inhibitor of the TRPC6 channels. So this demonstrates the capacity of mechanosensitive ion channels to sense mechanical force or changes to the cell environment and mediate functional effects. So how cells in a tissue engineered valve sense their mechanical environment is an important consideration, thereby determining what functional response they may produce. So we've seen that the valve shape and function are linked, and that the dynamic function of the annulus, the sinotubular junction, and the valve cusps are all linked. That valve interstitial cells and endothelial cells play a role in valve homeostasis in terms of either regulating valve tone or controlling calcification, and that alterations in the cell extracellular matrix communication and mechanotransduction may be key to maintaining valve function and durability. So if we go back and look at these key components of the biological aspects of normal valves and just speculate as to of these different strategies, which are important to which and how they might be best addressed. So within the in vitro tissue engineering approach, your choice of cells with which to populate your scaffold is a key choice. And we, in the early days of our project, did a lot of work in comparing mesenchymal stem cells, adipose-derived stem cells, their similarities to valve interstitial cells. And it turned out that adipose-derived stem cells were probably the best candidate. And for a while, we pursued this strategy. But ultimately, this is difficult to then translate this into an off-the-shelf product. And in terms of a commercial enterprise, it would be very difficult to pursue. The in vivo heart valve tissue engineering, again, here is dependent on your choice of scaffold. Fundamentally, since you have no control over what cells are going to enter your valve and therefore be implanted. To the more popular in situ tissue engineering, again, the scaffold choice is key, but also if you're going to populate your cells in order to get matrix laid down onto your scaffold, then this the similarity of these cells to valve interstitial cells is a moot point, so long as they do the job. So these need to be some sort of fibroblast-like cell, but ultimately you're again leaving it to the body to repopulate your decellularized valve. And again, in this term here, either you put this with or without being decorated by molecules or peptides in order to then either have a biofunctionalized or just a native scaffold, and this gets implanted. Again, here controlling the cell type you may have perhaps will rely on decorating the scaffold in an appropriate manner. So let's focus then slightly on the scaffold material. And while a lot of this is to do with the down at the bottom part of here about the nature of the actual material making the scaffold of, what you do with it may determine what cells will enter into the scaffold once it's implanted. So controlling the porosity and the fiber size and the alignment of the pores within the scaffold is an important consideration in order to determine what type of cells will get into your scaffold. As we mentioned briefly earlier, the modification of the surface or biofunctionalization of the scaffold, again, is an important consideration in order to achieve the correct type of cell attachment and migration and alignment in order to try to get a more valve-like profile of cells within your scaffold, and how these things also contribute to maintaining the mechanical properties of your material. So I now want to move on and briefly work through our current approach to the tissue engineering project that we have. And for us, we start with the native geometry and anatomy of the aortic root by taking CT scans and measuring blood volume area of healthy individuals. We've then derived via segmentation of those images into an idealized model of the aortic root. And our current project is aiming to produce a standalone aortic valve. The next step in this process is from that model is then to make this into a computer-aided design 3D model of the valve that can be then, the individual components of this can be 3D printed to produce a series of molds and dividers that allow us then to spray our scaffold material onto these molds. And this is essentially done by the method of jet spraying. So we jet spray our scaffold material, which is polycaprolactone, PCl. These fibers are dissolved in chloroform, then sprayed onto this rotating drum. The speed of the drum will define and determine the degree of alignment the cells will get, and therefore the porosity of the scaffold we make. And our aim was to achieve in these are called sinus molds, these form the cusps and the sinuses, highly aligned anisotropic scaffold material. These are then supported by a knitted structure that has a pattern that mimics the shape of the crown-shaped annulus, thereby giving the tissue engineered valve a similar, as best we can manage, mechanical profile of the individual structures of the annulus, the sinotubular junction, and the cusps, or the support of the sinus material, as the native valve would have. Then this is all put together and then solvent welded to bind the fibers of PCl together. And we end up here with a multi-layered composite aortic root, or the hair-filled composite component valve, as we now call it. So here's a picture of the whole thing, an aortic view, a ventricular view, and here's a nice cross-sectional picture showing the knitted area and the two outer nanofibrillar sprayed parts of the sinus wall, and then the highly aligned fibers seen in the cusp region of the valve. Well, I took this slide from a colleague of mine, and it did have a movie that played, but it doesn't want to play on my computer. And that showed a slow motion opening of the valve. And then here we have some echo images taken of the valve while in our bioreactor. And you can see the nice shape we have of the valve and a transverse and a longitudinal image. And this is from one of the earlier trials we did in sheep. And you could see that we got a nice cusp. There is some overgrowth here, which is an issue we need to think about. but a lot of particularly the knitted area, was rammed full of cells. We're in a process of really characterizing exactly which cells we have in here. So, as I mentioned earlier, one of the issues of implanting just scaffold material is you have literally no control over what cells enter other than governed by the porosity of the tissue. So one of the other projects we have, which hasn't yet married up with the valve production, but I'll share it with you anyway, is to decorate PCL scaffolds with this peptide, which has a great big long name, but is conveniently shortened to TPS. And this peptide specifically binds endothelial progenitor cells. So in an attempt to attract endothelial cells from the blood, knowing that they are an important component of heart valves, we set out to see whether this peptide could be bound to PCL. So we came up with the method of binding it by undergoing aminolysis, then malamide cross-linking, then cysteine containing TPS binds to these cross-linking molecules. And then if you can follow this side, so this is the different stages of the cross-linking process, arriving eventually with the TPS peptide bound. I know it doesn't look very good on my screen. You can see there's more blue dots in this panel compared to the others. But when we look at this by quantifying the number of cells binding at each treatment stage for comparing BOEX, which are stands for blood outgrowth endothelial cells. So these are endothelial cells grown from blood. So we can't really call them progenitor cells because they've undergone some change, but they are what progenitor cells, they would have started off as progenitor cells. And we compare those with valve endothelial cells, valve interstitial cells, and another endothelial cell line. And the peptide was able to capture significantly higher numbers of BOEX compared to any other cell type. So we need to work out how this technology can be applied to our tissue engineered valve in terms of how it relates to the production process and at what stage you could then realistically undergo this cross-linking pathway and add TPS to specific areas or parts of our tissue engineered valve. So this is my last slide, and it really poses the problems that everybody faces with tissue engineering, is that a lot of the results so far show this characteristic thickening of the valve leaflets and retraction of the cusps. And we've seen this thickening even in that 13-week sample I showed you. There was some over the growth, and controlling the foreign body reaction that certainly sheep seem to have, because this is what these valves have been implanted in, is one aspect. But again, the contractile response of the cells within these cusps seems to be pulling them apart. So trying to get the correct cell balance and the correct cells will be the key deciding step in order to get these sorts of tissue engineered constructs to function and work in the same way as the native valve, which is the goal that we are all trying to pursue. So I'd like to thank you for your attention, and I'm more than happy to answer any questions that you have. Thank you very much.
Thank you very much, Dr. Chester, for an excellent presentation. So we'll get right to several questions that have been posed. So starting with one from an anonymous attendee who's asking about, just to summarize the question, about what are some of the challenges associated with congenital heart valve disease in children, and how do physicians keep up with trying to manage critical congenital valve disease in children, and what are some strategies for kids with severe valve disease?
So the most common congenital heart defect is bicuspid aortic valve. So that affects between 1 to 2% of all live births. So those children will, all those people will at some stage possibly need some sort of valve replacement. So these people are good candidates for a tissue engineered valve. In fact, if anything, that is where most of people's efforts are aimed at helping people. So, you've got your 70-year-old, give them a valve that's going to last them 15, 20 years, it's probably going to be all right. If you're a 20-year-old or even younger, you don't want to be having valve replacements for the rest of your life. So that's where this is aimed at. And I think as tissue engineered valves become accepted, they can then be used in some of the other more complex conditions of congenital heart disease where valve replacements are required and needed. I know some people have congenital aortic stenosis. There are parts of the tetralogy of Fallows, I think it's associated with pulmonary stenosis. So there's a whole area of potential benefit where these people could enjoy from a tissue engineered valve, which if it's living can therefore grow with the patients. So I think that there is certainly scope for this, and I think that's where a lot of the effort is aimed at. I hope that answered the question.
Yes. So I guess to switch a little bit to the valve calcification in the context of, there's a question by Daniel Chaparro, who's part of Josh Hutchinson's group. So he asks a question about interstitial cells, and he wants to know, given the complexity of all the cellular components in the valve, and there is a wide utilization of in vitro assessment of wick function. Do you believe that, how confident are you about the results from in vitro assessment of wick function, given that wicks are predominantly fibroblast-like cells?
So I think that's actually a very good question, because a lot of people, including ourselves, have measured calcification responses in cultured cells. And of course, it's an artificial situation. And again, a lot of people, as we had done with the study with Jonathan Butcher, have used osteogenic media to induce a calcifying response. So that's one artificial situation on top of a chemical insult to try to induce a biological effect. So one of the things we've been doing with some success, although there's a bit of way to go yet, is trying to induce calcification in an intact porcine valve cusp with LPS, which stimulates an inflammatory reaction. And we are able to do this if you supplement the media with 3 millimolar phosphate. And I quite personally like the idea of this assay, because then you have cells in a valve cusp in their natural environment with the endothelial cells there and with the cell matrix there. So the next step that we're trying to achieve, and the pandemic hasn't helped, is then put that valve under stretch and challenge it with LPS and phosphate, and then have a dynamic model of valve calcification, whereby you're not relying on a cultured cell, you're not relying on beta-glycerol-phosphate and dexamethasone to induce your calcifying response. And you're able to see sort of what you might call biologically or mechanically stimulated calcification. And we've had some success in doing this in terms of we've measured the amount of alizarin red positive staining in sections of these valves. And we've also seen changes in valve cell phenotype in terms of more smooth muscle alpha actin positive cells and more osteogenic-like cells in the valve cusp that are stimulated with these pro-calcifying conditions. So hopefully that will be one way that we can properly try to address the calcification issue and then work out how to control calcification. And then that control of calcification should or then could be applied to tissue engineered valves, since it is more than reasonable to expect that they may well suffer from the same sorts of responses.
Great. So we have, I think, a couple of common questions, one from an anonymous attendee and one from Ms. Denise Su, who's actually a member in my lab, regarding the material selection for that knitted fabric that you're using in the valve, and is the formation of that knitted fabric, how is that fabric put together? Is it done by hand, or is there a machine that puts it together?
So I should have said it was it's made out of PCL. So we obtained some PCL yarn, and then we first went to a fashion designer who had a knitting machine, and that was sort of okay for making little square pieces. And then we got referred on to people with an industrial knitting machine, and they were the people who helped us make those jackets. And essentially, their machines, they were knitting shoes. You can knit any sort of object, apparently. So there were some challenges with the thickness of the yarn and getting it to knit properly, but we were able to reproduce any sort of pattern that we asked, or they were able to reproduce any sort of pattern that we asked for. So that's how those were made, and they were intentionally made of the same material that the rest of the valve is being made of.
So since Dr. Chester mentioned the word fashion, let me ask maybe a little bit of squeeze in a more broad question here, as in his role as he's currently the Deputy Director of Research at the Magdi Yacoub Institute. And I was just wondering, given a lot of the promises that stem cells and tissue engineering have promised, I guess maybe more of a philosophical question: how do you think we as researchers in the field of regenerative medicine can perhaps overcome some of the skepticism that has built up over the decades, stating that we're sort of saying more than we're promising more than what can be delivered?
I think,
how can we make it fashionable again?
Yeah, it's difficult. I think people in general have become a bit skeptical about science. I mean, that's no better illustrated by the move to resist people having vaccinations against the COVID virus. So quite why that occurs in society, I don't really understand it personally, but you're right, it's there. People are mistrusting. I think the only way to defeat that is to not lose our sight on what we want, what we're trying to achieve, scientific rigor in making sure we follow all the right steps. And ultimately, if we do reach our goal, the results will speak for themselves. If there are young people or children running around with tissue engineered valves in who don't have the prospect of being reoperated on, then there's nothing speaks louder than a medical success. So we will only be ultimately judged on what we achieve, and we shouldn't look to be judged on what we say. I think that's the only way to overcome sort of a skeptical public over what we're doing.
Great. So maybe we'll take another 2 or 3 questions. There's a question from an anonymous attendee about what type of assumptions do you make or you believe are most important when you are performing any calculations or simulations of flow through the valve? Maybe alluding to some of the models that you presented.
So I think that in terms of the cells on either surface of the valve, ideally, you should look to perform experiments whereby you can recapitulate the levels of shear stress and the patterns of flow seen on the aortic and the ventricular surfaces. So the machines or the flow bioreactors built by Philippe Sikorsky, his cone and plate machine, I think is the nearest approximation to doing that. However, you can then get caught up with the restraints that that puts on you in terms of how many cells and tissue you can measure. So there is room for having sort of a proof of principle with a more simplified system to show that cells respond differently to different levels of flow. And we've just done that with looking at the response, comparing the response of gamma interferon to TNF, a series of experiments that would have been impossible to do in a more complex system because the volume of work involved just would have been impossible to achieve the same number of observations. So I think you need to do most of your work in more simplified systems and then work towards using more complex systems that can mimic the flow environment that's seen in vivo on an in vitro basis. But ultimately, the biggest test is by implanting the valve into a large animal, which gives you that anyway. So in terms of a tissue engineered project, then that's your ultimate, or your one of the goals you need to achieve, and then you'll see what happens. So it's a matter of building up your level of complexity to suit the types of experiments you need to do.
So there's a question from Brittany Gonzalez from my lab, and essentially, do you have any results on the mechanical properties of the tissue engineered heart valve, and how similar are they to the native heart valve?
We don't have any results yet. So the initial study we did ended up not doing the mechanical testing to compare the mechanical profile to what you hope to achieve. We know that the valve is sufficiently strong enough to withstand durability testing in a bioreactor for a prolonged period of time. But we have another sheep trial which will finish shortly, which is a longer trial, and we will obtain the data that question addresses in the coming weeks. So it's a good question because it's exactly what we're doing.
Great. So, okay, maybe just 2 more questions to round up here. And I want to just remind students, both undergraduate and graduate students, that you will be having a noon session with Dr. Chester, where you can certainly follow up with additional questions at that time. So 2 more questions, one from Valentina Dargam from Dr. Hutchinson's lab as well. So she has a question about, I guess, going back to what Dr. Chester was saying about, trying to really get these type of products out there, and proof is in the pudding. I guess her question is on the scaffolds that are being used for tissue engineered valves, and what would be important criteria to determine if those scaffolds could move forward with human trials.
I think if you look in the literature, there are a whole range of different scaffold materials that are being used, various polymers. I think what is important is that you essentially want to end up with a valve that is entirely biological, so that, for example, the PCL scaffold is biodegradable. So the profile of your degradation needs to be slow enough to allow tissue formation, but not too quick. Otherwise, you won't get enough tissue formation, and your valve will fall apart. So the physical characteristics of its degradation rate are important, as is its inherent flexibility and durability. So there are various sort of hallmarks of requirements that are needed in order to realistically have something that's going to be able to withstand aortic pressures and flow once you implant it, because unlike any other tissue engineered product, the valve has to work straight away. There's no conditioning, there's no if you have tissue engineered bone, for example, you can, as long as if it's not weight bearing, you can slowly add weight to it progressively and let it condition to its own environment. With a valve, you get one go at it. So your scaffold material needs to have enough inherent strength to be able to withstand that. So we started off our project some time ago by trying to make collagen scaffolds, but we could never get them strong enough to be realistic that we could implant them, that we decided to abandon it and go for a polymer-based scaffold. I mean, you probably could have done it if you cross-linked it a lot, but then you'd have a relatively rigid structure that probably wouldn't be flexible enough to act as a tissue engineered scaffold. I mean, I may be wrong in that, because I know there's a group in Ireland that are making collagen scaffolds. I'd be interested to see how they do that and the basis for what they do it and whether they've implanted it. So there are various sort of minimum requirements you need of a material, but there's a whole range of different materials you can look at.
Okay, so I think to round up, we'll end with one more question, and I'll just read the question verbatim. This question from Henry Figueredo, and he refers to the drum method that was being used for the production of the artificial aorta.
Yeah.
And he wants to know, how successful is this method in terms of its repeatability?
It's been a what can I say? It's been a journey to make it repeatable. So when we started off, we literally had something someone had made by themselves on the bench from bits bought from the local DIY shop. And that's progressed to a excuse me, a much more complex computer-driven equipment whereby we can control whether the thing can spin very quickly. The control of the spin is regulated by the computer. The distance is set. The spray gun, which is there's lots of components, and spray gun is important. We now have a purpose-built spray gun and a syringe-driven delivery device to make the whole thing and reproducible enough to build valves that can be consistently used for in vivo testing. we have some failure rate, that has to be said, but the more we do, the better the system gets. And we're now actually trying to rebuild the machine to make it even more reproducible and more stringent and, if you like, user-friendly.
With that being said, I'd like to thank again Dr. Chester for his wonderful seminar and his really insightful responses to all the questions. And thank you, everybody, for joining us today for today's seminar. Take care.
Thank you very much, everyone.
Bye-bye.